Okay, quiescence is a major therapy challenge for cancer in general, but specifically for ovarian cancer. So quiescence is the induction of a subset of cells taking on a slow, non-dividing state. And that seems kind of paradoxical why that should be a problem. But all of our chemotherapies have been developed to kill fast-growing cells. Cisplatin, Taxol, they work by targeting cells that are growing very, very quickly. And that's why things like your hair falls out when we give chemotherapy, because hair grows relatively quickly. Chemotherapy kills fast-growing cells, so the hair falls out. And that's how chemo works, is because cancer cells are typically growing very quickly. But within the cancer, we know that not all cancer cells are created equal, and there are some cancer cells that are growing slowly. In fact, they may only divide once every week. And so rather than cells that are dividing once or twice a day, and they're going to see chemotherapy when we give it over a 24-hour window, those cells, if they don't divide during that 24-hour window, they're not going to die. And we think those cells are causing chemotherapy resistance and causing cancer to recur. They are particularly challenging to study because it's a little bit of the needle in the haystack problem. So if we take cancer cells out of a patient and put them in a dish to study them, and we take cells and 95% are growing rapidly, they keep doubling and doubling and doubling, and soon you've got 1 million cells in the dish. But those 1 or 2 cells that weren't growing very quickly, we can't find them because they are still 1 or 2 cells, and the other 1 million cells were all the cells that are growing quickly. So it is truly a needle in a haystack. If we can identify and target quiescence, it has 2 very important therapeutic implications. So first is, we know in ovarian cancer that 70% of patients, unfortunately, are going to have a relapse. We can treat them, their tumor looks completely gone. There's no evidence on a CAT scan or with their tumor markers, yet within about 18 months, 70% of patients are going to relapse. If we had a drug that could stop those quiescent cells, so what we think happens, chemotherapy kills the majority of the cells. There are a few of these quiescent cells left behind, and then over 18 months, they start to regrow. They make more of those fast-growing cells, and 18 months later, we have a cancer. If we could arrest those cells, if we could stop those cells from growing, if we could freeze them in time, and then instead of that patient's cancer coming back in 18 months, maybe it wouldn't come back, or maybe it would come back 6 years from now instead of 16 months from now. So that would be what we would call a maintenance therapy, a therapy that we could use to prevent those chemotherapy-resistant cells from growing. It wouldn't kill them, it would just keep them frozen in time, and a patient would go about their life. And in fact, we're starting to see some drugs, and we just had a clinical trial of a drug that works okay in this aspect, and it actually benefited 97% of patients who tested it in a phase 1/2 clinical trial. Now, the other opportunity, if we can identify quiescent cells, once we can characterize them, we could figure out how to kill them. So if we can kill 99% of cells with chemotherapy, and then I can come up with a drug that can kill those quiescent cells, now maybe cancer doesn't come back. And that's really the ultimate goal, and that's the holy grail, is can we take those 70% of patients who aren't cured, and can we target those quiescent cells such that they now will die and not cause cancer recurrence? And so that's the primary goal of something we're working on. So by studying these cells, we were able to collaborate with some colleagues where we could literally take cells and put 1 cell in a dish at a time. And we could do this with thousands of cells at a time. We had a what's called a micro well, where we could put 1 cell in a well for 10,000 cells. And what we found was there are about 5% of cells that weren't growing. They weren't dying either. We isolated those cells compared to the other 9,995. And we looked at them and found what was different. By doing that, we've been able to infer a therapeutic. And so we have actually now discovered, I can't call it a drug because I can't use it in patients yet, but a compound that we're hoping will become a drug that actually, when we treat ovarian cancer cells, very aggressive ovarian cancer cells from patients, when we treat with that drug, they completely stop growing. They completely arrest, and they go into what we call a quiescent state. And we can characterize that biologically, but essentially they stop growing. So if you put those cells in a mouse and you treat that mouse with this compound, the mouse doesn't die of cancer. Cancer cells don't go away, but they just stay there. They don't grow, they don't die. If we stop the drug, they'll grow and kill the mouse. So that's exciting, and that's something we'd love to be able to do for patients. What's even more exciting is that by characterizing cells, we've found what I think is an Achilles heel. So these cells are dependent on a certain way that they eat proteins, and they use those proteins to stay alive. And so there are drugs that are already clinically available which target some of these cells. And so now if we take our new compound and we combine it with the existing drug, we can actually take those quiescent cells and kill them. And so the idea would be if we could move this into a patient, we could actually prevent cancer from ever coming back by killing those remaining quiescent chemotherapy resistant cells. So the big picture is that we can use this compound. We could treat patients with chemotherapy. Residual chemotherapy resistant quiescent cells are left behind. We could target them to force them to not grow. And then while they're quiescent, we can add a second drug and actually kill them. And that would lead to tumor eradication or cure. And that's the big picture. That's the goal. Is I think we can use this approach to actually increase cure rates of patients, which is what I've been striving my whole life for. And so that's the big picture. That's the goal. I can add as a simple analogy to understanding quiescence, you can think of the hair follicle. And so you say, well, why the hair follicle? So in our hair follicle, hair is fast growing, as I mentioned at the beginning. Chemotherapy kills fast growing cells. And so hair falls out. All of our patients who get carboplatin and Taxol chemotherapy, all of our ovarian cancer patients will lose their hair. But all of them, 100% of them, the hair will grow back. And why is that? Because in the hair follicle, there are quiescent stem cells, and those cells do not get impacted by chemotherapy. So the hair always grows back. When the chemotherapy washes out, the stem cells can start regrowing again and cause the hair to grow back. And so I think the same thing is happening in ovarian cancer, where there are residual quiescent stem-like cells, which when the chemotherapy stops causes the tumor to grow back. And we're going to target those, we're going to get rid of them, and we're going to increase cure rates for ovarian cancer.